institución. Actualmente es el jefe de la colección y también es jefe de coordinación del Departamento de Agricultura de Estados Unidos, la sección de Florida. Ha sido galardonado en 2009 por la Sociedad Entomológica de Florida, premio al logro de la entomología reguladora. En el 2018 recibió el premio Edward Berger, Departamento de Agricultura, y un premio a la trayectoria por su compromiso profesional, experiencia científica y la innovación en el campo de las ciencias vegetales reguladoras. Esto es para la sección de lo que es como fitosanidad equivalente a México. Ha publicado alrededor de 130 artículos científicos, diversas revistas, todo sobre taxonomía e historia natural de escarabajos, principalmente sobre Erotílidae del Nuevo Mundo, escarabajos que son exclusivos, bueno, no exclusivos, pero la gran mayoría están en los hongos y polinizadores de cícaras. Y los escarabeidos afodinos, que son escarabajos, digamos, en su estricto peloteros. También es editor de la revista Insecta Mundi y fue coeditor de los volúmenes 2000-2022 de American Beetles. Su interés por la investigación se alimenta en la emoción y el descubrimiento, investigando los hábitos inusuales de los grupos de escarabajos que tienen sus líneas de investigación. Es especialista de ambos grupos a nivel mundial, lo cual, y puedo decir que personalmente Paul Skelly es realmente una persona bastante accesible por todos aquellos que tengan interés. Adelante. Em, el día de hoy Paul nos va a presentar una charla bastante interesante, espero que sea de su atracción, y es todo un ecosistema complejo que ocurre en un hoyo, básicamente. Paul,
You.
all yours. So, I guess this is no batteries.
Yes, it worked a minute ago. Okay. Greetings. I think you read the title, but I'm here to talk to you today about the Tuza and the special ecosystem that it sets up. Now, as Adelson mentioned, I study certain scarab beetles, and these scarab beetles are associated with this rodent. So if any of you are studying any organisms, you know, it's very important to know about the biologies of the other organisms that yours associate with. So today we're going to talk a bit about the Tuza. The Tuzas are a special group of subterranean rodents that infrequently come to the surface. They create burrows in which they live and they hunt their food matter, the plant roots. They're primarily vegetarian, and they also use the plants from the surface for bedding in the nests. Right, so their bodies are cylindrical, okay, to be able to run in the burrows real easy. They have a sparsely haired tail because they run backwards as fast as they can run forward. The front legs, great big claws for digging. They have reduced eyes and ears because they really don't need them in the burrows. Large front teeth for chewing roots and tubers and to dig through the soil. But really cool about this group is that they have a fur-lined pouch on the outside of the mouth. Other squirrels and things like that have a pouch on the inside. That's how these get some of their common names. Okay, the family of rodents, Geomyidae, occurs throughout North and Central America, and they have different common names depending on where you're at. In Mexico and Central America, they're called Tuza, which comes from the Nahuatl named Tozan, which sort of means ground squirrel, moles, things like that. There may be other regional names that I haven't heard about yet, but I'll leave them out there. Right now, there's 4 genera and about 10 species in the region. In North America and Canada, they're referred to as gophers or pocket gophers, again, in reference to that pouch on the outside of the mouth. There's roughly 3 genera and 31 species. Now, I want you to pay attention because it'll come up later. Right over here in the southeastern US is an isolated species that we studied. I'll talk about them in a minute. They're called sandy mounders because of the habit of these rodents when they make their burrows to push mounds of sand onto the surface. But also, whoops, whoa, that was interesting. Also, there's a real local name where they're called salamanders in reference to the mythical creature they're born of fires. And they get that name because living in their burrows, wildfires can go through an area, volcanoes can devastate the landmass, but these rodents are underground. They have their food caches, they feed on roots. they're frequently the first signs of life that you see after a wildfire goes through an area. And one of the reasons that they're important, to get the right button, is that when wildfires go through an area, they can kill the seeds that are on the surface. And so, but this rodent burrowing will then bring seeds back from under the soil, bring them to the surface so they can start regenerating and rejuvenating the habitats. So these little rodents are very important as a keystone species in many different habitats for different reasons. Habitat restoration, food for predators, and in prairies and grasslands, they can benefit the soils by keeping the soils churned up and soft. Otherwise, they'll get compacted. And if any of you worked in areas of grasslands, you know the grass roots can get so dense that water can't penetrate. So these rodents' activities of burrowing is helping maintain the grasslands, the soils profile, and all sorts of great things for the whole habitat. Also in their burrowing, they'll be bringing leached nutrients, all right, that leach down in this. They'll bring those back up to the surface to help the plants. They're also burying plant matter that creates more organic matter and fertilizer for the plants. And interesting that these sand masses also act as germination sites for pioneering plants. So if you have these rodents in the area, you frequently have a greater floral diversity. In human culture, they have multiple uses and places they show up. Entertainment, there's popular kids' books with gophers as subject matters. They actually, I found this, I didn't even know about that. They dig so well, they have garden tools named after them. I found them as names for restaurants. There's actually in the US a place called the Tuza Taco. I don't know if it's because they look like a Tuza or are they made with Tuza. We'll find out. But also, I was surprised, but not, there's some regions where they actually eat them. Not yet. I was hungry enough as a graduate student. I almost did. But they are best known as a pest. The mounds are considered damaging to lawns and pastures. The burrowing can disrupt garden plants, the tender roots, seedlings, things like that. And they can feed directly on the plants. They've been seen to burrow be in their burrows, grab a plant and just pull it under and then feed on it. And the plant just keeps ratcheting down as the rodent's feeding on it. So in gardens, they're considered a pest, in pastures. And so much of the information we know about these rodents and their life histories is based on them as a pest and ways that we can then potentially control them. I'll just point this out briefly that in some of our research, we're also seeing that even though they're a pest in many systems, they're also kind of disappearing in others. So in some places are in decline. So as a keystone species, I've already mentioned, they're very important to habitat health and restoration. But what we've missed mostly are all the other animals that live with them. First, everybody knows about ectoparasites, fleas, lice, mites, all those fun things. Those are studied by people that are working on medical veterinary things. Also, these animals, these insects tend to have co-evolved with the gophers. So there's a lot of people that looked at the evolution of both the gophers and these insects and all sorts of fun stuff. I'm not going to talk about any of that. As I mentioned, these rodents burrow. And when they burrow, they push the sand up to make these mounds that are so obvious when you're driving around. These mounds create habitat for other animals. We have found snakes and birds basking on these mounds in the winters. I've found snake eggs, lizard eggs, things like that actually incubating in the mounds. These mounds act as nesting sites for other insects, things that might need a little bare patch of sand to nest. The burrows, when they push the sand up, it buries whatever organic matter is on the surface. As that matter rots, other insects feed on that. There's some group of beetles that I worked with in Florida. That's where you find them. You don't find them anywhere else. The larvae are feeding on that decaying plant matter. Oh, and germination sites for plants. But what is really exciting is the burrow itself. It is an ephemeral cave. The rodent is constantly modifying it, moving it, but it's always open. And there's a lot of different insects that have specialized into this cave. So the burrows are home sweet home for the rodent. Has everything you can want. It's closed. They don't like to keep their burrows open. So when they come to the surface, when they're done, they'll typically plug that burrow to protect it from the surface weather, from predators, and any other sort of intrusions. It's only opened when they desire to have it open for mounding or for drying tunnels out if it gets too wet. They have a kitchen, a pantry, a food stash where they store their food. They have a nest chamber full of warm bedding materials to go hunker down, sleep through the bad weather. They have a latrine for proper hygiene. They're not dirty rodents at all. They're really clean. They have deeper sections in the burrows so that if it rains too much and water seeps in, the water will run and drain away so they don't get flooded. Oh, and this is the fun thing. See the spiral? At least in the southeastern US, the rodents all have this spiral in the burrows, and it's suspected to be a way to escape predation. If a snake gets in the burrow or if some other mammal gets in the burrow, the rodent can quickly zip through there, go off, the snake or other things gets confused and gets stuck. And so while it's stuck there, these rodents can quickly then backfill the burrow so that they're gone and safe. Really special little guys. What else do we got here? Oh, yeah. Tender tips of all the plants that they've cut through will grow back in. So those could be little snacks for the rodent or additional food for the insects. Oh, and the constructions, these are rodents after all, they do what they want. The construction has a lot of these commonalities, the different rooms, but how and where everything happens is different. It all depends on the soil structure, the water drainage, soil profile, the materials it's made out of. This last little bit, in sandy soils that are easy digging, we, and I say that literally, we have dug burrows that are over 60 meters long and 4 meters deep. Now that's big. I don't recommend that. Most of them are not so. The soils tend to be more compact and they don't dig quite as far. Okay, so I mentioned it's a cave. So it's like a cave in that these rodents bring in all the nutrients, the plant matter, from the surface into the burrow. They feed on it, they defecate. Whatever's left over might then begin to rot. So there's all these nutrients in this cave, on which then there's coprophages and saprophages feeding. And then you got a whole wealth of predators feeding on those. And of course, you got big coprophages, little coprophages, all these. I mean, it's a network. It's a complete network. So, Tuza burrow fauna. There are special inculin insects that have to have two adaptations to survive in the burrows. First, they have to be able to find the burrow. Once you find the burrow, you're golden. Man, it's temperate, it's humid, the food is brought to you. What more could you ask for? But the trick is getting in there. The second trick is to live with the host. Imagine you're trying to live in a subway tunnel and the train's coming down the pipe. What do you do? Okay, that's what's going on here. You've got this little tunnel, and all of a sudden, here comes this road that occupies most of it running through. How are you going to get away? So there's special adaptations frequently some of these insects have to deal with that. We can have morphological adaptations, which was very intriguing. Long, skinny legs. You can go to the side and hang onto the side of the burrow as the thing runs by, or even on the surface, on the top of the burrow. You can have chemical defenses. A few of these insects reek. I mean, oh my God. The only thing we can think of is it must be a rodent deterrent so that the rodent then stays away from the bugs. And then you can have behavioral adaptations. I haven't observed it, but I read about the camel crickets. Everybody's seen camel crickets, big, lonky things in these burrows. Rodents come down the burrow. You feel the breeze. Where do you go? These crickets have a behavior that they'll build a side tunnel. And so when they feel the cricket come or the gopher coming, they'll run into the side tunnel. just like the subway station, subway tunnel. You either hang on to the walls or you find a way to get out of it. So all these insects have a different way that they manage to survive with these rodents. All right, some of the different groups. We have Diptera and scarab beetles that are the primary detritivores. We have things like small mammal nest beetles that might feed on the molds and fungus that's growing on all this organic matter, or maybe they're feeding on the mites that's feeding on that. Camel crickets, what's surprised to me is that they're also predatory. So they're feeding on some of the other insects along with the plant matter, all sorts of predatory beetles and all sorts of other taxa. Hey, there's moths in there that are feeding on the nesting materials too. Pseudoscorpions, mites, just all sorts of exciting stuff. And then you get the accidentals. Hey, you're underground. Some of this subterranean fauna, these really bizarre things, accidentally will break into the tunnels and live there for a while. Well, I was told I shouldn't talk too long, so we'll just keep moving on. How do you sample these burrows? Like anything, you got to make sure you have the right permissions for any area you plan to typo. Anyway, any areas you want to collect, what permits are needed, what aren't needed. Some of these insects do come to the surface to disperse, but that's very brief, period. So you can collect them on the surface by all those different methods. But if you collect them on the surface, how do you know they're associated with the rodents? You don't, unless you already know it's an associate and this is what that thing is. So it's cart before the horse. So we don't gain that much biological information except in knowing how they disperse. If it's a pest in an agroecosystem, we found that frequently the land managers are more than happy to let you work with their gophers, to remove them or just study them or do whatever, dig up their fields, discourage them. So we found that for us, the best methods are actually collecting from the burrows, which means you're going to get dirty. You can get like a stick with a can or a kitchen ladle on it. Don't use your mom's ladle. To scrape out the rodent burrows when they're open. Or you can actually open a burrow and start digging. And just follow that burrow and feel it until you find the nest in the dung chambers. That way you can also find larvae and you can rear them and you get a better idea where in the burrows these different animals live. But because you're doing the manhandling of all the material, you miss a lot of the tiny things. So what we have generally done is went with removing the rodent from the burrow and setting a pitfall trap in it so that then all the little animals that are running around in there could be collected and you can see the total fauna. As I mentioned, some of the physical limitations. What technique are you going to use? Well, first, are you digging in volcanic soil? Are you digging in sand? Are you digging in the prairie where the root mass is so dense and hard to dig that you can't, or in a forest? So we need to be familiar with all these different techniques because every area is going to be different. Rocks and boulders. Yeah, I dug under a boulder once. That was interesting. But the trapping method is we think is relatively easy. You open the burrow, you remove the rodent, and there's many methods to do that. Then you set a pitfall trap in the bottom of the hole you made with the burrow, cover the hole with the board and sand. Now you do that because if you don't cover with the board, then all these surface animals will come in. So you want to know what's in the burrow. So you cover it to keep the entire burrow factors, the humidity, the temperature, all that the same so that the animals are running around, but also to keep the surface stuff from gaining entry. Then you check it a few days, a week later, and remove the catch. So this all started for me and Dr. Kaverick, your next speaker, in the early 1990s, and we were both living in Florida. And we had started getting a little interest because there was a couple of beetles that we realized were with these rodents. And we thought, well, look, there hasn't been much done. Let's look at these in more detail. So we selected it because, well, first of all, like I pointed out earlier, one species isolated in one area. So that was good. We just knew everything is going to be associated with this host. And we also had the work of Hubble and Goff. In the 1930s, they surveyed burrows in an area of Florida and produced a phenomenal work. If any of you want to read about that whole ecosystem and things that are going on, that's the work to go to. Since then, nobody else has done that sort of ecological survey. And so then, yeah, we dug into the literature, we dug into museum records to plot our different maps. You can read that and see you can see there's some decline even in the southeast, some places where we don't think they occur anymore. But the other thing we did that was interesting is that we selected a locality and collected samples from there every month of the year. That'll become important later. But our favorite groups are scarab beetles and Histerids. That's what we focused on, but we collected everything, and most of it we've shared with other specialists who are working on it, or it's still sitting in my lab in a cabinet. I did not do that. No, I did not. Stop. Okay, so time? You watching my time? Okay, good. So just to keep this short, Hubble and Goff, the foundational work reported 2 species of scarabs in the burrows in Florida. We added 10. Hubble and Goff reported 4 species of Histerids. We added 5. Now, okay, let's back up a second. Why are there so many species in a group that does the same basic things we think, ecologically? Dung beetles, how much dung is there? How are they stratifying that niche? We got too many questions, folks. I can't answer that one, but they're there. All this fauna is there doing something different. So after we finished our multi-year project, and we're still publishing, what did we learn from this? Well, first, that Hubble and Goff's foundation level work was incomplete. They only sampled a small area of Florida, so they missed fauna from other places, and they also surveyed it in late spring. And they missed parts of the fauna. The burrow morphologies differ depending on the communities, which I mentioned before. The burrow fauna, even in the same area, can differ. And the only things we can think of is the age of the burrow, the age of the rodent making it, luck of the draw. Did a certain group of insects manage to break into that burrow to be able to colonize it? So don't just think you go to one place, you get a burrow sample, you're going to have everything. No, it doesn't work that way. You need to really work it to understand the fauna. This was the exciting thing we finally realized, that the distribution of the insects is not the same as the Tuza, the rodent. It differs. And what we started seeing was different soil types. Certain species preferred certain soil types with the burrow, or this other soil type with the burrow, or even climatic things. In Florida, the peninsular Florida basically has summer all year round, but farther north, you get a winter. So in the peninsula, we get a different fauna than we get in the northern part of the range. So there I just spoke with it. We've got the seasonality. Why the seasonality? The biggest thing that was really exciting too is that these rodents estivate. Most rodents hibernate. So these Tuza, they become active in November and they're working all winter long, and then they sleep in the summer when it's hot, dry, and the soils are so hard you can't dig them anyway. The insects follow that behavior. When the niches and the things come open, then the insects are active. More attacks than expected. Some fancy side stories. Every taxa has a different story to tell. With the camel crickets, after we got the species, got them identified by the expert and started plotting maps, we realized each of those species is not separated by a soil type, they're separated by a river. And more excitingly, is each of those species, the closest relative is in the prairies in the western US. So all these distributional questions are things that we just, like I said, that's another seminar. So then we expanded into the western US, and more taxa, more effects. This group, I know my time's coming up. We found out, like I say, winter active. That group is flightless, really rare in collections. Why? Because it's living with the rodents under the snowbank. you got to go out in the winter and sample on the snow to find these things. Keep going. Shifting interest into Mexico. We did the same sort of thing. We tried to pull the literature together, see what's going on. And it's mostly scattered works. A little here, a little there, a couple species here and there, except for the genus Onthophagus, where Halfter and Zunino and then Sanchez Huerta have been working on that for over a decade, getting bits of information only on that genus. And you can see, interestingly, one species is widespread and common, but then you just get just outside of that a little and poof, you got new different species. Why? Who knows? So we jumped in, got one of our colleagues to help us look at Querétaro. 2 localities known, we sampled them different times of the year, same fauna both times. One mountain range had described species that were known. The other mountain range, they were undescribed, completely new. More work needs to be done. More things need to be answered. I've skipped some things where I talked about the transition zones. I mentioned the faunas in the US changing as you go west. It changes as you head south. And so this quick chart, you can see it visually, that up here we have, we have the genera listed and the number of species by region. And you can see that here in the eastern US, we have these genera. You move to the west, you kind of shift down to these genera and look at the number of species. This will be important. And then you move over here into Mexico and you're getting more of these genera. But pay attention to the totals. We've got a lot more to find in Mexico, folks. And then what about this? I keep telling people, if you can get into a Tuza burrow south of Mexico, I can guarantee you're going to catch new species. It's just going to happen. We have no idea what's down there. Regional endemisms also. Conclusions. Sorry, 5 minutes? Hot dog. Okay. Tuza are an important part of many natural healthy ecosystems as keystone species beyond what we generally understood for maintaining those ecosystems. Tuza are more common and widespread than our literature implies. I skipped over it, but in Querétaro, on one of our last trips, we started asking the local people, well, have you seen any Tuza? And yeah, they're everywhere, up and down the mountains, but all that was in the literature were these 2 localities. So if you want to work with them, talk to the locals. Let's see, where are we? Faunal activity periods can change. In Querétaro, it's like here. It's very temperate all year round, right? You get some moisture, you don't get some moisture, no biggie. But if you get into lower elevations where you have a hot dry, where the soils dry out and get really, really difficult to dig, the rodents apparently do the same thing. We were in Jalisco last year and we could barely find them. They were there. We did find signs of them, but they were just shut down. They were inactive. Guess what? The insects would be also. If you do manage to study these and you're working in that niche, which is a difficult niche to break open and work with, catch other taxa besides what you're working on and share them with specialists and other people so we can start as a group combining our knowledge on these things to get a bigger picture. If you're unable to do studies like Onthophagus, where they looked at this whole region, or like we did in the southeastern US, you looked at the whole region, well, pick a small region and work it hard. Up and down the mountain, different elevations. Look at soil type differences. Look at the faunas you get. Look at seasonality. I mean, a study of just that one region would be very beneficial to help us understand what's going on in these systems. Mexico has many genera and species of Tuza, many natural systems across a very diverse landscape with lots of wonderful phylogeographic history that just can account for so much more diversity than what we're currently seeing. Although we have already seen US into Mexico, faunal shifts, transition zones. We don't know what's going to happen with a lot of these birds, but we're expecting a lot. Mexico is going to be rich in what it has. So these systems deserve a lot more study to discover the treasures they hold. Thank you. Yep, you shut it off. I had an acknowledgement slide. Okay. Yes, 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 yes.
Yeah. Okay, so I have a question,
And I hope I have an answer.
I mean, you said that there is like some affinity between the population and its Coleoptera diversity as well. But my question is if it is possible to characterize the population of Tuza depending on the community diversity of Coleoptera that inhabits.
To characterize the Tuza on the road, the insects.
The populations depending on.
You mean as if as if there's a healthy beetle fauna, therefore you have a healthy Tuza population?
No, like if it is possible to characterize the specific Tuza population, if you have the information about the composition of the Coleoptera diversity.
I'm thinking the answer is no, because what we're seeing is that the insects are, these burrow insects are following the burrow, not the Tuza. So in the Western US, there's all those different species in different genera, and you can have the same beetle species in multiple species of Tuza. They're associated with the niche. Yeah.
And it is related to the previous question. I mean, you said that there are some beetles, like that one that is like specific for winter.
Yes.
The cave system. Hola. If my question if the cave system is similar to endemism hotspot or something like that, like a micro level.
That's
no. My question is this motherfucking. I hate this.
Okay.
Ok, so mi pregunta es si él mencionó ahorita que habían escarabajos que eran específicos para ciertas, no sé, como temporadas X. Y no sé si en estos bichos, o específicamente para los sistemas de cuevas de estos ratones, uno puede hablar de procesos de especiación en una escala micro, o si uno puede llamar esos sistemas de cuevas como islas de endemismo.
So, how I think if there's a expert specialization it will be associated with the whole system of the process, and if you can use those, like sample to related of diversification of the beetles based on the two samples.
If you can look at the beetle diversity, and also well, we're still learning what's even there. For an example, in the Southeast, we had 13 species of scarabs. Other regions, we only get 6. So far in Mexico, we're only seeing 2 or 3. So that's why we need to, I think, first of all, get some good solid information of what sort of diversity are we looking at in different age regions before we can start making predictions. Because if we were to use the Florida model to predict what's in Mexico, man, we're way far behind. But if there's only like 4 genera of scarabs in the burrows here in Mexico, as opposed to a dozen, in Florida, we're still gathering information, I think, to be able to answer questions like that. I hope that helps. Yeah, I mean, this is a plea for help. Yeah.
Thank you for the nice talk. It was very amazing to see also the rodenter. I want to ask about the insects. Do you find them also like free living outside the, what's the name of that? Burrow.
The burrow, generally no. They do come to the surface when they disperse to find a new burrow. Because remember, this system is moving, right? So you're a larva, of a beetle, you're in the dung chamber, and all of a sudden the Tuza decides it wants to move house, right? So it then buries that whole burrow system and creates a new one somewhere else. So you emerge as an adult beetle, you got to find a new burrow. So you come to the surface, and for however you manage it, visual, chemical, whatever, you then fly and find a new burrow. I mean, that's typically a very brief window. There's a season in the prairies in the US we call Indian summers, when after that first rain in the fall, the temperature, the soils are wet, and then the temperature warms up just a little bit, okay, before winter hits. That 2-day period, the wind stops on the prairies, and these beetles from these burrows will fly in such dense clouds you think it's snowing. But if you're not there for that two-day window during the day, it's only in the afternoon. So they have these very brief periods that they disperse. So that's why sometimes you might get one accidentally to light or a flight intercept, but they spend the majority of their time underground with the rodents. Thank you.
Si existen más preguntas para Skelly, al final con todo gusto puedo responderlas. El siguiente ponente es Peter Kovarik. Peter trabaja en el Departamento de Biología, Física y Ciencias Matemáticas en Columbus State Community College. Obtuvo su grado de doctor en entomología en 1994 en Ohio State University, Columbus. Es maestro de ciencias en entomología por Texas A&M. Y desde entonces hasta la actualidad, pues ha trabajado arduamente en un grupo específico de escarabajos. Es profesor de diferentes cursos en la misma institución, Columbus State Community College, en los cuales da principalmente lo que es temas de inducción a la biología y entomología. Es curador de la colección de insectos de Museum of Biological Biodiversity de Ohio State University, Columbus, Ohio. Ha sido también a su vez galardonado con el segundo mejor póster en algunos eventos. Alguno que es bastante destacable es el Snodgrass Memorial Research Award, que fue en el 96. Entre otras de sus actividades y la cantidad abrumadora de publicaciones que tiene, bueno, en 2020 recibió financiamiento por un proyecto en el cual pues tuvo oportunidad de visitar y ver todos los tipos para esta familia a nivel mundial. En sus diferentes estudios ha viajado a muchos países de América Latina, desde Argentina, Brasil, República Dominicana, Guatemala, Puerto Rico, Venezuela, y de hecho el que tiene más viajes en su vida, alrededor creo que son como 36 de todo lo que hemos hablado, México. Absolutamente, yo creo que él conoce más nuestro país que nosotros mismos. Bueno, Peter, please. Should I leave the Tuza here? Next back. Next back.
Greetings, everybody. It's good to be here. Paul was talking about essentially accumulating data, and it's tough to do that when you don't live in an area. But we're doing our best to try to unravel some of the secrets that are here in Mexico and get students involved in these projects who live here and can help us out just adding pieces to the puzzle. So that's something that we're anxious to continue. And I wanted to just point out this picture here. This is an SEM from a book that I bought very early on when I was studying Histerids. There wasn't much written comprehensively on Myrmecophilus and Uzihalava and what did that happen? All right. Okay. Yeah, it wasn't you. There's ghosts in here. So yeah, Uzihalava, Henry Howden, and Al Ritchie put together a publication which had SEM micrographs of a lot of these Myrmecophiles. This is eye candy to me. I went nuts. You see something like this, and it really, really hooks you into a particular group. And that's partly why I decided to work on Histerids. So go to forward here. Okay, there we go. All right, so there's a beginning to this. So Histerids are a very small group of ecologically distinct predaceous beetles, and they a significant number of them live with ants. And there's some pre-adaptations that allow them to do that. But with regard to data accumulation, this all starts back in the late 1800s. Then an ambitious project was started, which I'm sure everybody's aware of. That's a biologist that down here. Biologia Central Americanae was conducted. And this ambitious project was set out to document all the flora and fauna of Mexico and Central America. And they got a lot done. And George Lewis was he was in charge of the Hysterid chapter for that major work that came out. And 129 species of Histerids were recorded in the Biologia. And of that number, only 7 of these were Myrmecophilus. And so subsequently, a lot of sampling effort has gone on, and a significant number of new country records have been documented for Mexico. So let's go to the next slide here. Oops, it's going the wrong way on me. Oh, I just want to point out too, that illustration right there, that comes from the Biologia. The illustrations were superb. And for the most part, those illustrations allow you to identify to species what they found way back when, instead of having to go and see the types. But it's always good to do that as well. All right, so this shows in red what Lewis published, and these are subsequent. The taxa in black are things that have been found since the Biologia was done. So it's not too shabby. 5 subfamilies, 38 species of Myrmecophilus. And the co-author on this talk here, Alexei Tishetskin, he went all over the world and got data on Myrmecophilus. Most of them occur here in the neotropics. And all that data was painstakingly put into a database that I was unaware of until like 2 weeks ago. When he finally shared this information with me. He's a Belarusian, a very, very stoic, stubborn person. I get along with him, love him to death, but sometimes he's really slow in helping people out with important information. I mean, this is a treasure trove, and it really shaped what I'm going to be talking about today. And there's so much more that we can learn as we accumulate data and add it to this spreadsheet that I now have at hand. So at any rate, going forward with this, megadiversity, I mean, this is the place to go. If there's ever a place to come and work on Hysterid beetles, this is it. That's why we come down here. This is well worth it. Every reason in the world to come here. All these biogeographic different zones here. You've got 2 major North American and neotropical faunas converge here, and you've got this transition zone, which Adelson told me is now recognized as a special unit. So this is just a great place to do field work. And one thing I want to emphasize here is, because this is such a mega diverse area, every time we come down here, we just get overloaded with material. And we've got to learn to be a little bit more targeted in terms of some of our endeavors. So another real good reason to work on Myrmecophilus Histerids here is because pretty much all of the ant genera that host Histerids. You've got Atta, Eceton, Labidus, Nevomyrmex for the to begin with, and they've got Formica, Phodoli, and Solenopsis. These all occur here in Mexico, and some of the numbers of species for these genera is just incredible. All right, so let's move on to the next. Come on. Oh, there we go. Okay, it's a little bit slow. All right, so Myrmecophilus are essentially social parasites. This is like going to a bar with a friend who doesn't bring his wallet consistently. They just take advantage of the hosts. So they live totally at the expense of their hosts. And there are few insect orders that have produced Myrmecophilus. And among those that have are Coleoptera, and it's the staphs and the Histerids. These are the two beetle families that have produced the most Myrmecophilus. And they've got some things that are some of their behavior ties into their morphology. So if they are not living with the ants, if they're living at the periphery of an ant colony, chances are pretty good their morphology is not going to be that modified, and they're going to be recognizable as just a general Hysterid. They are not super modified. But if you are socially integrated, then your body's going to be bizarre, like you can see here. They're very, very strange looking. And they share some things in common, those that are well integrated with the ant societies. So they are small in size. Look at how tiny this thing is. They've got grasping notches, the arrow's pointing to one. Those are little handles that the beetles, the ants grab onto the beetles with those little handles, and they carry them around in their nests. So that's what they're doing. And also, they've got trichomes. Why do they have trichomes? They've got glands that secrete substances. We'll get into that a little bit later. But substances are secreted, makes the ants happy, and so they are carried around in the colony. But remember, these are worker ants. They're females. All they do is work all the time. They're sterile too. I mean, it must be lousy to be a worker ant. So they entertain themselves with these little toys that secrete drugs. And they have fun with this. Okay? Get it? That's what this amounts to. And the ants are getting something out of this relationship as well as the beetles. So expanded tibiae too. So a lot of these beetles have expanded legs so that the ants can also carry them around by the legs, should they choose to. Okay, behavior. This is pretty cool. So the bodies of the beetles, again, that are poorly integrated, look like normal Histerids. Those that are highly integrated do some crazy things. They do some crazy things. Those specimens that are associated with army ants travel with the colonies. Army ants, I don't know if you've experienced them, you probably have at some point in time. If you're a biologist, you're out in the field, you see these things. They run, and they run fast. And these histers keep up with them running, or they do something kind of strange. This guy is riding on an ant, like you would ride a horse, except these guys face backwards. Don't ask me why. They do that, okay? So you can see one running along with a colony here. You notice the Hysterid in that picture right there. You see him? They call it the butt beetle. It's clinging with its mandibles, the Fungidium. I'm fine. The ant. And they just cling to that area and ride along. So all this stuff, I love animal behavior. I mean, there's nothing better than animal behavior. And when you get into this, and it was sublime morphology. So another thing about Histerids that I absolutely love is their morphology. They are organic. They are works of art. And that's why I love to take pictures of them, because a picture's worth 1,000 words. These are pieces of art that belong in a museum. The problem with these beetles is they're too tiny to appreciate unless they're magnified. And I bring boxes of insects to my class. They're all big things so that students can appreciate them. But it's under a microscope that you fall in love with these beetles. All right, so we go forward here. Oops. So passive trapping. When Biologia was written, nobody was running flight intercept traps or barrier pitfall traps. So a lot of the diversity was missed. And this is partly because the beetles generally don't go far away from where the colonies are. So that's a limitation there. But by running flight intercept traps, we picked up a lot of Myrmecophilus. And barrier pitfall traps do a decent job of this as well. And active sampling techniques. This is a pain in the ass. It truly is, because the ants aren't happy with you digging up their nests, and they bite you and they sting you, and it's really, really rough. And a lot of times there's a learning curve, and sometimes you pay a price for that. Like Atta colonies, if you mistakenly, you're trying to find their underground garbage dump, and you mistakenly dig in their nest chamber, they will chew you apart. They will shred your clothing. They'll make you, and then your friends see what's happened to you, and they make fun of you as well. So it's a lose-lose situation for the entomologist. Just letting you know about that. So this is me in Belize digging up Atacephalotes years ago. Digging up Atacephalotes nest, and it took me a long time. I got some great stuff from there. And wonder who this character is right here. Do you recognize him from an earlier talk? So yeah. This is a disclaimer. He comes along these trips. He collects more Histerids than I do all the time. And it aggravates me a little bit, but I come to grips with that. And I benefit so much from the deal that I don't care anymore, that he collects more than I do. This is one of the world's best collectors of beetles. I'm just letting you know that. That's meant from the heart. So anyway, it's tough work. So there are 3 species of Atta here in Mexico, and 2 of them have got underground garbage dumps. One has their dump on the surface, and that makes it easy to find stuff. That's what Paul is working with right here. Atta mexicana, surface debris, easy to find the beetles there. Cephalotes and mexicana, not so easy. You have to dig up the nests, and you risk severe body damage by so doing. But this isn't half, excuse me. Here we go. Yeah, this is the worst active sampling technique. It's only been done 6 times in Mexico, and there's a reason for that, because you wear protective clothing. Sometimes that protective clothing fails. There's some problem with it, and the ants get inside, and then you are wearing a cage that's full of army ants that are stinging the crap out of you. And it's not fun. It really is not fun at all. So why do this? Why do colony sampling? First of all, it tells you what the host is. Sometimes there are mistakes, but generally, the Histerids beetles that associate with ants are very, very host specific. So we learn. That's another piece of the puzzle that we can add to the whole, the collective. And so it's kind of worth it. I've been on the losing end of this as well. And this is Tabasco. I got into some army ants there, and this is last year in Jalisco. And notice, Addison over here taking pictures. Look at how far away he is from the action. And he's smart. He's staying away because he doesn't want to get stung and bitten. And I'm close without my protective gear, helping Paul out, and I paid a price for that as well. But the adrenaline rush that you get from doing this is nothing like anything else I can suggest. But it's worth it. It is so worth it, because you get a load of material, you have to process all of it, but you find host associations, you find all sorts of things that you wouldn't find just by scanning FIT pans. All right, so Ataphilic histers. So you've got a bunch of them that occur here, and most of them are in the dumps, where the leaf scraps are discarded. So that's where you find most of these histers, and they look like normal histers. So this is Plagiogramma pubifrons, a hidden species. Look at the face on that thing. Isn't it kind of cute? Pubifrons, that's where it gets the name from. I get a kick out of that. The males have got that feature. The females do not. They have an interesting courtship, which I won't get into because we don't have the time right now. But at any rate, real cool ataphile. And there's another one here in Mexico. The type's missing. There's not a single species that is known in science. We went looking for it yesterday, and we failed because of the heavy rains that have been happening where the type locality is. But we'll not give up. We'll keep looking around. And then Pseudister rufilus. All right, so this is a exosternine. This is one of the few exosternines that get into Atta debris, And they're rare. They're rarely collected. I feel lucky that I've collected a few of these beetles here in Mexico, and I think they're beautiful. All right, Hister. This is a big genus overall, and there's a lot of histers that get into adder debris. And you've got a couple of the rare species here, but you also have common species. And it's not surprising that 5 of the recorded species of Hister were found in the Biologia, because people were sampling out of surface dumps, out of Mexicana surface dumps way back when. And Epiglyptus costus, this was thought to be a Mexican endemic, and we've since found them in El Salvador and Guatemala, so no longer restricted to Mexico. But this is a cool beetle. I got to rear this thing. I brought back live specimens with a permit. I was able to rear this. Haven't described the immature stages, but I've got them preserved. And this is the only dendrophyline that associates with Atta, Zystopygia multifarianum. There may be another species here. We don't know yet. All right, and this is Geomysoprinus formicarius Hinton. This is the other thing we went looking for, because Hinton's description was so crappy that we wanted to make sure that this is what he was finding in this place south of where was it was Teopilco? Oh, Teo. Okay. All right. So at any rate, and Acritus species. All right. There's one species, one Attaphyla that's been described. There are many species that occur here in Mexico and throughout the neotropics. None of them are described. So this is going to be a nightmare for somebody to work on someday. And this is the only Heterion. This is the only one that's really integrated into the ant society. And this is found in the fungal garden of Atta. And beautiful thing. Okay, Solenopsis, another ant that you don't want to mess with because fire ants, bad news, bad news. But you've got some Histerids that are found associated with their nests, and that's a big surprise. But you only have 2, and that's not so surprising. And let's just take a look at the first one. Come on. All right, so this is Hippuhister castaneus. Described by Lewis from Mexico and unknown localities, not given on his label. And finally found one in an FIT when we were doing our work in Tabasco. Found one in Belize too, and that was a new country record for this beast. And Wenzel described a species from Veracruz, Cordoba, and it's gone missing. So this is another thing we want to get maybe to Tuxtlas and see if we can find some more of these things. The illustration is fantastic, so it's not going to be a problem to say that we have this thing. But at any rate, this is one of those that associates with Solenopsis. And this is the other one. Got one in a FIT in Querétaro. I was just really floored because these things are not commonly collected. The genus was revised not too long ago, and it was too late for me to include my Querétaro record in that publication. All right, and then we move on to my favorite. Look at these. And the indigenous people in Brazil, they use these ants to stitch up their wounds. That's what they do. They just they let the mandibles sink into the wound to close it. Then they just pull their heads off and leave the heads in their bodies. So that's kind of interesting. What? Okay, he's warning me. All right, so we're going to haul through this. But I want to we're closer to the end than you believe. But all right, so there are the neat thing about these guys is they raid. So they have swarms that raid all over the place in the daytime, and they just they kill, they dismember, and they drag back to the colony all sorts of things. And it's thought that the Histerids are feeding either on the ant larvae or the booty, what the ants bring back, or a combination of the both. And the Aceton burchellii is probably the most common host of most of these Myrmecophilus that you'll be seeing here. And they have a biphasic lifestyle. So they've got a stationary phase and they've got a nomadic phase. All right, so the stationary phase lasts 3 weeks and they stay in the same area for all of that time. And then the nomadic phase, they set up a different bivouac at a different place for a 2-week period. So that's kind of neat. It makes it tough to find the appropriate phase to sample. Anyway, this is a real ant that demands respect from virtually anybody. Even Don's birds do track, some of his birds track these ants, right?
Yes.
All right. So anyway, this is a new genus near Aleodentodes and Holodytes. All right, we're not so sure about the host. We're suspecting it's associated with the eceton, but this has been demonstrated to be in something that's part of Mexican fauna. Chilista lucidus, right, real cool looking thing too. This is probably the most common, even though it's not been reported. It's probably one of the most common Myrmecophilus that has been found when you've done colony sampling. And this thing, got this in Tabasco, and I paid the price for that. But the closest position or closest location for this genus is Brazil. Yeah, so that is incredible range extension. Jose Luis collected this in Chimalapis in Oaxaca, not reported from Mexico. So this is a beautiful beetle. I got some of these in Belize, got some finally here in Mexico. This is a real cool thing. Again, Tabasco. This Tabasco is a great place to go looking for Myrmecophiles. Euclasia, another beautiful little beetle here. Clientister. Remember the poster for this talk. That's what I chose because they are just so freaking bizarre. They really are. Recently revised too. This is just one of many that are just incredible. Aphanister, fungifer. All right, all of the rest of what we've been seeing so far, they are mostly going to be associates of Eceton burchellii. This is associate of another Eceton that's here, Eceton hamedum. There's 2 more Eceton species here. We have no idea if they host any Histerids. All right, Labitus. Two species of Labitus. These guys tend to be more subterranean than superficial. And there are a slew of Histerids that are associated with this group. And this is one of them. And then we've got Neva myrmex, much smaller species. They are active at night. And this is why you tend to pick up the Histerids that associate with them at light, because they're out tracking the ants at nighttime. So they come to light. So that makes it easy to find them, but you don't have a host association necessarily. And this is Alkeus. These come to light. None of these are described. There's only one described species in the whole genus. So yeah, you can yell at me because I'm not keeping up with things, all right? And Paul is probably just not too happy either about my progress on getting publications kicked out, but so be it. I got other things to do, like love my cats, okay, Paul? So deal with it. All right, so Formica, 2. There are tons of species. I know, I'll be done with this in a second. There are tons of species of Formica in Mexico, much more than the US, and so few Histerids are known from Mexico. There has to be a lot more. So we expect to find more Histerids here in Mexico. Badoli. These ants have got workers, some of them with large heads, and they're just very, very bizarre. Another area of megadiversity is Mexico for this genus. And this is Terapus. Okay, this is one of the associates of Fadoli. And I'm supposed to have this thing described. I'm working on it. And check this out. These are the hind legs that the ants are going to be clinging to move these things around and pulling. These are all glandular trichomes. So that you have glands that are secreting substances into these channels here, and then these hairs are picking them up and wafting the substances. So these are getting the workers high. These are walking beer kegs or walking stashes of marijuana. I don't care how you want to think of this, but these are drugs that are appeasing the ants. They're toning down their aggressive behavior. So this is just really cool stuff. And this is another Fidelia associate pine exister. All right, we got a bunch. Well, not a bunch. We have some here for which the hosts aren't known. This is one of them. We don't know what the host is. 2, we don't know what the host is, both known from Mexico. And this crazy thing was recently described. There is a closely related species of this beetle that was reared from a tarantula egg case in South America. So they may be, these species that occur here in Mexico may be feeding on tarantula egg cases. We don't know yet because nobody's done any kind of rearing. So how do they find that out? Some people were sneaking egg cases of tarantulas into Europe to raise them for a pet trade. And instead they got beetles in the egg sac. So anyway, that's how we know. All right, what's next? A ton of shit to do. Need I say more? Okay, I'm being hurried up here. But yeah, we have a lot more to do, a lot more information to accumulate. And to show you what we have done, that pubifrons, the fuzzy face, this is the type locality. And that was the only place they knew that it occurred for years. And now we've got a bunch more places. So this, again, data accumulation. That's what we're trying to do. Increase the amount of information we have on a particular group. And shows you some of the new things that we've turned up here. So at any rate, on to the acknowledgements. Thanks are due to a lot of people. So particularly Robert Jones here and Adelson. I come down here. They are hosts, gracious hosts. They put up with me and my garbage. And they're like brothers to me. I don't have any brothers. I got 2 sisters. I hate them both. But at any rate, these guys are salt of the earth. Very, very much cherish what they've done in terms of making me feel well accommodated, help me with the translation. I speak a little Spanish, but not enough for me to even attempt to talk like this. But at any rate, a lot of thanks are due to some of the people who have done some collecting here, helped us out when it came to dealing with these army ants. And photography was largely done by Paul and by Jeff Gruber. He did the auto montage work for this this talk, and also I was in touch with Alex Wild, who does this incredible ant photography, and he allowed me to use his images without having to pay for them. So I was real thankful to that. Should I show the shot of Robert with the chicken? All right. Oops, I shut it off. He saved you from the.
¿Tenemos tiempo para preguntas?
All right. En español.
Hola.
¿Qué tal?
So I just saw like there is certain factors in morphology in all these groups, and like you explained that these parasites of legionary ants have like long legs and that sort of things. And my question is if cultural modifications differs in Histerids depending on the habitus or the range of movement on the ant species.
Yeah, I mean, you have a lot of Histerids that ride the ants, and their legs aren't modified like you saw on those that were running along with the ants. So those that run have very long legs, just like the burrow Histerids. They've got elongate legs just to be able to Paul, move very, very quickly and keep up with their hosts. If they don't keep up with their hosts, there's a price to pay. So that's, you tend to find those features exclusively in those ants that are running along with the, so if you live underground in a Phydoly nest, you don't need long legs for running. But a lot of those morphological features that you're talking about are strictly associated with army ants, which move rapidly. And these beetles have to keep up or they get lost. So they can fly and pick up the colony odor and then drop and do it that way. But usually they run along or they ride the ants to just maintain communication with them.
And my second question is related to that one. For example, let's suppose that you have let's say, all the social parasites of an species. It is possible to identify that ant species depending on its parasite community, or it is not possible?
No, it's going to be possible. We have that database I was just telling you about, that spreadsheet from that Alexei got all the data from all over the world in terms of the collections. Once that data is really critically analyzed, that should be telling. We should be able to predict on the basis of the Histerids where the ants are found and vice versa. That there's such an integration, such a level of integration. It's more host specific, less niche specific than what Paul talked about. So there's such a tight communication. It's necessary too, because you have to live in that community. And if you don't disguise yourself, you're persecuted. And I should have gone into this a little bit more when I was starting the talk. The adophiles live away from the nest, and they're not persecuted by the ants. The ants are pretty good at keeping things at bay that they don't want in their colony. But these Histerids are wolves in sheep's clothing. I can't speak Spanish well, but if I could, you wouldn't think of me as a pinche gringo. Maybe you think of me better. But it's just like that. These beetles, they speak ant. They speak ant. The ants don't know that they're there, and yet they're eating their babies, getting them high. I mean, it's just fascinating. I mean, it's just cool stuff. What's not to love about this? And these beetles are just you saw how bizarre they are. Like I said, they're little organic artwork. So anyway, I think this is just so neat, and this is a great place to be. Thank you guys for coming to this talk. Love Mexico, I really do. I've been here so many times, it's not funny. One of these days, I'm learning Duolingo Spanish. I took it in high school too. I swear, one of these days I'll be able to communicate and give a talk in Spanish, I promise.
Si alguien tiene preguntas en español, con todo gusto podemos apoyarlos. Bueno.
Yeah, thank you about your talk. It was amazing. The photos, all beautiful. But I was wondering, do you show a species that inhabits in the fungal gardening? Do you think they also eat mushrooms?
Pogonomyrmex?
I'm not sure. I didn't.
The seed. Yeah.
Fungal garden.
The fungal. There was one.
Yeah. Do you think they also eat the fungi, or they are just
No.
there?
Maybe. Who knows? If the ants can eat it and get away with it, who knows? It may be that possible. We don't know. We do not know. And you're the first person that has ever asked that question, and it caused me to think. So I'm going to say possible, because all right, think of it this way: the immatures of the atta are supposed to be feeding on protein. So there has to be protein content coming from the fungi, at least some. So that might be sufficient. A lot of these Histerids feed on fungal spores. Not most of those that we encountered today, but the group I work on, they're fungal spore specialists. That's protein too. So that is a great question, and that's something that needs to be looked into. Thank you.
Some of these beetles, the ants eat them.
Yeah, they do. They solicit food. They do this with their front legs, and then the ants feed them. They act like little babies. They stand up on their hind legs, and they do this with their forelegs, and they get fed by the ants. It's just cool. Reminds me of my cats when they want to be fed. They do all sorts of crazy things. All right. So, at any rate, thank you very much.
Bueno, agradecemos su presencia. De igual manera, reitero, cualquier pregunta con todo gusto van a estar aquí disponibles. Hoy todo el día están en el instituto, bueno, en la colección, pero cualquier cosa que necesiten, adelante. Hoy, por términos logísticos, no hay café. Entonces, pero voy a pasar a hacer la entrega de constancias. Foros. Yes. You need to take it. Bueno, voy a leer, ya para terminar, solamente hago la entrega de las constancias. La primera constancia dice, el Instituto de Biología de la Universidad Nacional Autónoma de México tiene el honor de otorgar el presente reconocimiento a Paul Skelly por la impartición del Seminario Extraordinario y a su vez también a Peter Kovarik por impartir también un seminario extraordinario. Entonces, guys, up there. Just put in the middle. If you wish.
Well, if you get a
Thank
photo,
you.
yes, thank you. Good night.
Oh, okay. No, that one. Hey, take your carpet. It's supposed to be there, formality. This looks like a person, muchas gracias nuevamente. Maestro Jorge, ¿cómo apago el micrófono? Ok.